Hello, good afternoon, all of you. Are you ready? Okay, thank you, all of you. Okay, let me start my lecture. So I am going to speak on nucleic acid binding proteins. So many of you may have heard about various types of nucleic acid binding proteins. So there are two things in my lecture title. One is the nucleic acid and another is a protein. And most of you in this group know both are biomacromolecules or biological polymers, nucleic acid being constituted by polymerization of nucleotides, and proteins being uh, constituted by the polymerization of amino acids. We'll not go into the basic chemistry of the polymerization, but these are all kind of primarily thread-like structures, but they have higher levels of organization. And nucleic acids, to begin with, are naturally occurring chemical compounds. And all of us know they serve as the primary information carriers in living organisms as well as virus. We are including virus as a separate entity because viruses are on the borders of living and the non-living. And you know, composition of the nucleic acids vary depending on whether it is a ribonucleic acid or a deoxyribonucleic acid is having a unique combination of these bases, particularly uracil in RNA and thymine in DNA, and the rest of the things are similar. Now let us go to the protein molecules about whom we are going to discuss how these proteins bind to the nucleic acids. So as I already told you, a protein may be composed of hundreds of or thousands of the building blocks, the amino acids. They are very large complex molecules and do most of the work in cells and are required for the structure, function, and regulation of the body's tissue and organ. And most of us consume a lot of protein every day. And those who eat eggs know that when you boil the egg, the white part, which is basically the protein, which exists actually in a liquid state in the raw egg, becomes hardened. And that's actually basically a phenomena called denaturation. So proteins behave in very different ways when exposed to temperature or pH conditions, and there are many other things. Now, these primary chain of amino acid, as you can see on the left-hand side, they will actually fall into a higher order structures. They turn into a coil-like structure, which we generally refer as the alpha helix, or they may form in a planar structure, two-dimensional structure, which we call as pleated sheets, basically the beta sheets. Now, these helix and the pleated sheets may arrange with respect to one another in three-dimensional space to give the tertiary structure of the protein. And when a protein is composed of more than one polypeptide chain or more than one polymeric chain, the arrangement of these two polymeric chains or three or four polymeric chain with respect to one another gives the quaternary structure of the protein. The functionality of a protein basically lies in the tertiary structure and the quaternary structure. So this concept is useful to us because when we say nucleic acid binding proteins, the protein has to be functional. So if it is in the primary structure, it's just opened up like a rope, it won't be able to carry out its function, not only in nucleic acid binding, but also other functions like catalysis or etc. Now, one very basic concept, all of you know, we have four bases due to the codon usage. They represent 20 different amino acids, right? So this is the basic concepts in biology. We have around 20 amino acids, and these amino acids, as I already told you, constitute the protein polymers or polypeptides. Now, question arises: the four bases represents around 20 various amino acids due to the triplet codon. You know about the codon dictionary. Now, how many different kind of proteins are there in this world, which are, produced by a combination or permutation of these 20 amino acids. If somebody knows the answer, you can type it in the chat box. But of course, this is not very easy to answer. Now let us go to something called the PDB, or the protein database. So if you look into the statistics of this particular database, the first protein structure was reported in this particular database around 1976. So there are two things: the total number of protein entered and total number of structures released per year. So in 1936, they reported 13 protein sequences in this database, and all the data, all the 3D structures of these proteins were solved. Now I took this data from this RCSB website and compiled this graph by using the data every 10 years. And you can see every 10 years, there's a huge jump. Initially in the first decade from 13, it became 213. But the number of structures solved didn't increase in that way. From 13 to it increased only by 18. Now, by last year, and in fact, this year itself, you can see this huge number of protein being reported in this database. So how many proteins in this world? It cannot be estimated as of now. This is likely to grow more and more. And this slump may be not because there are less number of proteins in this world. That may be because of policy issues, or even maybe the COVID impact, by the year end this will go up, or other priorities we have. But what is interesting is this part, the number of structures released annually. So last year they released around 14,000 protein structures. This year they have already released around 8,300 structures. So this is what our knowledge about protein overall is in this world today. You know about the central dogma of molecular biology. Those who don't know and are from other disciplines, but may have studied about it in your high school, let me give you a revision of the concept. Our genetic information basically is stored in the double-stranded DNA molecule, which do not do any work itself. It is just an information repository. Now, to carry out any kind of work, for example, expression of a color in a flower, or some flavor in a fruit, or some eye shape, or some kind of a fitness character, DNA, the genes in the DNA has to be expressed. That is done by the process of transcription, which produces basically a blueprint for the protein. And these RNA will be actually further translated. So RNA is one kind of a language. DNA is a language. The language of DNA is deoxyribonucleic acid, and the language of RNA is ribonucleic acid. But now, this nucleic acid is entirely getting translated into another dialect made up of amino acids. So that process we call as translation. And those who are experts in molecular biology, you know how this translation process takes place. Now, this flow of information in a way is unidirectional from DNA to RNA to protein, until and unless it was discovered that there are RNA viruses where the genetic information is not there in the DNA molecule, rather they carry RNA molecules as repository of information. But to again express themselves, this RNA has to get back translated into DNA by a process called reverse transcription. And then finally, that DNA again gets back translated into RNA, and finally translated into protein. Now, in this entire concept of central dogma, you have three players: the DNA, RNA, and the protein. But amongst these, DNA and RNA, they mostly act as information carrying molecules. But the major player here is actually protein. If there will be no proteins, DNA cannot be transcribed into RNA, and RNA cannot be translated into protein. Even for viruses, the RNA molecule cannot be reverse transcribed back into DNA. So you see, protein actually drives the central dogma of molecular biology in a way. So if you consider the concept of genetic information as a will passed on to us by our ancestors, protein act as the executor and trustee of the genome. Now, what I want to emphasize before moving to the next slide is that our topic of discussion is nucleic acid binding protein. In this slide, you have nucleic acid on one side and protein on another side. So for doing all this work, whether transcription, replication, yeah, I forgot to tell you about the replication process. This is a process by which DNA makes copies of itself. So even DNA replicating itself, making copies of itself, it requires the assistance of protein. Now, in this entire process, protein has to interact with DNA and RNA. So whenever there is interaction, protein has to bind to these molecules. That is why we are going to discuss today about these particular things, nucleic acid binding proteins. So by this time, we were now all convinced that proteins bind to nucleic acids. Okay. So what are these nucleic acid binding proteins? We have so many different kind of proteins within yourself, and I showed you the number of protein structures sold last year, 14,000. This year already 8,300, right? And a huge number of proteins already reported every year. Now, out of these, there are only a few proteins or a percentage of proteins which are capable of regulating genes by interacting with DNA, RNA, or both respectively. Now, to do this, these nucleic acid binding proteins act in a cooperative manner with each other and perform the vital functions in every stage of gene regulation. Now, for example, if you see here, if you look into the general function of nucleic acids, some of them act like transcription factors. I told you about the process of transcription, how RNA copies are made from DNA molecule. So you can see here the DNA molecule. So in this DNA molecule, you can see big globular structures. These are basically the proteins. So these proteins actually bind to the double-stranded DNA and drives the process of RNA synthesis known as the transcription. So you can see here a small pink stretch, we call it as the target gene. So this gene will be transcribed and produced into RNA. So this process, you see the role of protein in a way. Now, there are other proteins which bind to RNA. So these RNAs, when protein binds to them, so they result in some very interesting phenomena inside the cell. Some of them we call as alternate splicing. We know that mRNA contains both the exons and the introns. Exons are the part of the gene which will be finally expressed in the protein, and we call them as exteins. And introns are actually thrown away or discarded in the process by a process called alternate splicing. So to carry out these alternate splicing, the proteins have to bind to the RNA. Now, mRNA molecules are very unstable. We know that once the protein binds to the mRNA, it provides them stability and also save it from getting degraded. Now, there are other functions like, for example, certain proteins will bind to DNA and RNA at the same time, or they may use certain modules which one of them binds to the DNA while the other binds to RNA. And you can see here the non-coding RNA, one small molecule is being taken, which binds to this RNA binding protein, which forms a complex with the transcription factor, which binds to the DNA, and the non-coding RNA binds to activators. This is a very complex geometry or assembly, as you can see. This is what we were speaking about, that cooperativity between the nucleic acid binding proteins. So once these complex assembly is done, in this case, transcription will start, right? And this is much more complex than the first case, where simple transcription factors bind to DNA and immediately starts off transcription. It's, of course, again, not very simple. There are many players associated with it. But compared to the first case, the third one is much more complex because you have here other players like non-coding RNAs over there. Now, apart from these, the nucleic acid binding proteins may act in another mode. So here for this fourth case, you can see there's a long non-coding RNA which binds to a DNA and RNA binding protein, and then on the one side, and kinase on the other side. So this kinase also has RNA binding motifs in it, and this kinase will actually phosphorylate the DNA and RNA binding protein by this process, by transferring the phosphate group to it. And this leads to the activation and sometimes deactivation of the particular protein. And this can lead to the transcription of a target gene. So this is, in brief, the different modes by which somebody is telling me if we can use pointer. So yeah, I'm trying. Let's see. Is the second slide moving? Yes, sorry. Okay. Now let me go to another database, which is known as the Eukaryotic Nucleic Acid Binding Protein Database, or the ENDP. If you are interested, you can actually visit this site, which I am putting in at the bottom. Now here, the group used roughly around 1,770 available transcriptomes in the database, and around 790 genomes of eukaryotic species. And you can see the huge number of 28,66,000 nucleic acid binding protein getting predicted. And this was distributed around 2,423 species. Now, before we go to the next concept, let us make ourselves clear that many proteins bind selectively to various substrates. In case if you are unable to hear anything, please send some message in the WhatsApp group also. I am keeping an eye on it for your input. Okay. So the proteins may bind to DNA. The proteins may bind to RNA, and the proteins also bind to themselves. So there is a protein-protein interaction and protein DNA or a protein RNA interaction. Now, we are already aware from the earlier discussion that nucleic acid binding proteins fall into three different classes. One is DNA binding proteins. Okay. Another is the RNA binding proteins. So these proteins will specifically only bind to either DNA or either RNA. But there are some members of proteins which can bind to both DNA and RNA at the same time. We call them DNA and RNA binding proteins, or DRBPs. Now, if you look into some examples of some model animals like Homo sapiens. Homo sapiens is not a model animal. You cannot do direct experimentation in that way. But of course, we do a lot of genetic sequencing studies on humans. But others are model organisms, the Drosophila, and then, Deniodarea, etc. So you can see the number of total nucleic acid binding proteins printed in red on the extreme left. And you can also see the ratio of the RNA binding protein to DNA and RNA binding protein, as well as the transcription factors listed over here. Now, let us go to the next concept. What would happen? What would happen? Rajiv Kangavan, you can type in the WhatsApp group whether my voice is still audible, so that I can keep a track that everything is going on fine. Okay. Now, what will happen when proteins and RNA interact with one another? So this may happen in two ways. A RNA binding protein will act on RNA molecule. So this is the first case. A, you can see this yellow structure representing RNA binding protein, and you can see the small hairpin in blue, which is a small RNA molecule. So in this case, the yellow molecule protein is going to bind the loop RNA. So when this happens, it will facilitate processing of the RNA or modification, or it may provide stability, and it may also, in other cases, lead to translation and even localization. So it will drag the protein molecule or the RNA molecule inversely to certain locations. So this is what happens when an RNA binding protein act on a RNA. Now, what happens when a RNA will act on a RNA binding protein? So here the picture, the player is reversed now. RNA is going to bind to the RNA binding domain of the RBP. So these also trigger certain things. It will give some kind of a function, attribute some kind of a function, or help in some kind of an interaction, as you could see in the earlier case where that long non-coding RNA or the non-coding RNA was providing some kind of function. And then other things like stability and localization will also be facilitated. Now, this particular course, we are discussing a lot, or we're going to discuss a lot on the genome editing and genome engineering technologies. There is a complete lecture on CRISPR-Cas9, so I won't really tell you here something in detail. But here you can see this particular principle of RNA and protein coming together and doing some work. Okay, so this CRISPR-Cas9 system basically relies on two components. One is the RNA, the another is a protein, which is basically a nuclease enzyme. And this RNA we call as a guide RNA. Okay, so you see this yellow structure is the Cas9 nuclease protein, and this hairpin green and gray structure is the RNA molecule. We call it guide RNA because without these RNA in conjugation with this Cas9 protein, the Cas9 has no any kind of directionality. I mean, it's a very potential person, but he doesn't know what to do. Cas9 can still maybe carry out the function, but it will be not very specific. It may just go and cut any DNA here and there. But attachment of this RNA on this protein, okay, gives it some kind of directionality, and it will drag it to its DNA target molecule. So you can see in the lower picture here how the RNA guides this Cas9 to the particular DNA target. So that's why you call it as a guide RNA. So this is just to illustrate this particular point B. What happens when RNA act on a RNA binding protein? So this is, I mean, something magical happens, and this is a tool which won the Nobel Prize last year. So I leave it to the next speaker to really tell you about this very interesting topic. Let me go on with the basic fundamentals of this particular discussion. Right now, the story of RNA binding is over in a way for the time being. Let us move on to the class of molecules, nucleic acids, which bind to both DNA and RNA. Now we look into both the Venn diagrams on both the sides. So one on the left side is being done by generated through a low throughput experimental evidence, the other on through a high throughput studies. That's why you get different numbers over here. In the first case, they had a total set of 588 DNA binding proteins and 1210 RNA binding proteins. Then they found out among these set, there are around 64 members which bind to both DNA and RNA. In a later case, they found out around 407. So this is a report from 2014, but probably this number has increased in the last 7 years. So this is just to show you the ratio of DNA to RNA binding, but it depends on the type of study or the type of experiments being carried out. But you know that there's a common set of proteins which both bind. There's experimental evidence which bind to both of these. Now, I spoke to you about a RNA binding to a protein molecule, taking CRISPR-Cas9 as an example, and what wonderful magic it can do due to that. Now, if you ask me, what are the purpose of these proteins which bind to both DNA and RNA? I mean, are they do they carry out something specific, or they are confusing? A lot of studies has been done. In this particular study, around 149 candidates were taken. Okay. Now, who is telling try to reconnect? It's working fine. I think everything is going on fine, right? Okay. So you see, out of this assessment of these 149 candidates, they found out it plays diverse roles like positive regulation of transcription, cellular response to DNA damage stimulus, mRNA processing, telomere maintenance through telomerase, regulation of cell cycle, regulation of apoptotic processes, DNA replication, then response to heat, viral infection, and then other processes like DNA methylation, and so on. Even response to cold as against response to heat. So these are the various functions of these DNA and RNA binding proteins, very, very diverse and multiple. Now, what are the various structural properties of these kind of proteins? So these DRBPs have RNA recognition motif, then nuclear binding, nucleotide binding alpha beta plate, then nuclear hormone receptor ligand, and this is the distribution you can see in this graph, and many others like K homology, zinc finger nuclear hormone, then Kolsov protein DNA binding, zinc finger NHR gatotype, then double-stranded RNA binding, then helicase super families 1 and 2 ATP binding. So these are the various kind of structural properties these DNA and RNA binding proteins have, for which they recognize the target DNA or RNA partners. Overall, there are three archetypes of DNA RNA binding protein function. Number one is that in this case, RNA, you can see this decoy RNA can compete with DNA for binding to DNA and RNA binding proteins, typically at the same protein interface. In the case of transcription factors, this can reduce promoter occupancy and the transcription of target genes. So here, the favored site is same for both of them. So that's why sometimes RNA will try to replace DNA by binding it to it. And you can see this is a bidirectional reaction. So this helps in switching off and switching on genes. So whenever RNA transcription need to be stopped, so these particular protein DRBP will be snatched away from the system by the DRBP by deploying a RNA molecule. So thereby detaching it from the DNA conjugation. So very clever of the cell to utilize this particular protein having dual function. So this secondly, these DRBPs regulate gene expression at multi-levels. In addition to binding to the promoters of genes to regulate the transcription, the DRBPs can also affect microRNA processing, as well as mRNA stability and translation. So, as in the first case where it does some very simple job, here you can see it actually is involved in multiple and multi-level operations. So, the third case is these DNA and RNA binding proteins can bind DNA and RNA simultaneously, allowing the RNA to function as a scaffold to recruit other proteins to a specific DNA locus. So this you already see in the case of C in the earlier cases. So here you see this SF1, here this red colored one, SF1 binding to a long non-coding RNA. You can see this RNA with the many hairpin motifs. Steroid receptor RNA activator SRA to recruit the SRC1 transcriptional complex in a ligand-dependent manner. So all these players are required, and then these will start the transcription. Now let us go to another important topic. So you now know something about the RNA binding proteins. You now know something about the DNA and RNA binding proteins and how they work either alone or in competitiveness with one another, or in a cooperative way, right? Now let us go to one topic which is known to you, the topology of DNA. And you know that DNA exists in a double helix form, right? Its diameter is 20 angstroms. And then for every turn or pitch, in the ideal BDNA, you have 10 base pairs, which is about 34 angstroms. And over and above, what is important for our discussion today are the minor group and the major groups. So DNA double helix structure contain two groups, which are known as the major group and the minor group. Now, many of you may be already knowing why this major group and minor group occurs. Still, I will highlight that point once more to you. Something we need to remember that the major groove is wider and deeper than the minor groove, and you can see the figures over there: 12 versus 6 angstrom and 8.5 versus 7.5 angstroms. So why does these groups arise? They arise because you can see the glycosidic bond on the left side and on the right side of these base pairs. So these glycosidic bonds of a base pair are not diametrically opposite to each other. So these groups result as a outcome of this kind of arrangement. And these groups are lined by potential hydrogen bond donors. You can see them in colored in blue, and acceptors, which are colored in red. So these are some of the information we need to carry with us. Now, we know that DNA, there are proteins which binds to DNA. And probably all of you are also aware that there are some classes of enzymes known as restriction endonucleases, which can recognize DNA sequences, right? If somebody is facing connectivity problem, am I audible? Meeting is not getting connected. Sergi, Meenakshi, maybe it's an individual problem, or it's, am I audible to all of you, right? Anyway, so yes, sir. Okay, thank you. So now you can see the possible base configurations in here, 4 possible base configurations: GC or CZ, AT or TA. And here, just to show you, in all the 4 cases where the major group and the minor group lies, and these are the hydrogen bonds between the bases and the hydrogen atoms attached to carbon, which are unavailable for hydrogen bonding. So they don't have any role in the protein interaction. And then there are potential hydrogen donors, bond donors, and acceptors, as you already know, and methyl groups, which form hydrophobic protuberances. Now, in certain cases, DNA actually is recognized from the edges, from the outside, without opening the double helix. So you may not be able to understand at this moment these particular concepts if it is you are exposed to it for the first time. But it is not always that like the transcription factors which go and binds to a data box, or the restriction endonucleases which go and bind to consensus sequences, right? It is not always required that that sequence specific binding is there. Even the DNA can be kind of identified by the protein by some kind of a touch mechanism. So we will discuss what that touch mechanism we are. Okay. Right. Discussing about. So we already know that these nucleic acid binding proteins play major roles in cellular processes, and DNA binding proteins are no exceptions. So whenever a protein, here you can see in the ball structures, field structures binding to this DNA. Now what happens when a DNA binding protein binds to DNA? So it starts the process of transcription that we already know. It helps in the process of DNA replication, and also DNA packaging or packing. And it can cut DNA, which you call as DNA restriction, or it may repair the damage that is being done due to any processes of mutation, or even some kind of replication error or whatever. So protein will bind to the DNA to do the repair. And of course, crossover events like DNA recombination. So all these reactions depend on DNA binding proteins binding to the DNA molecules. Now, how do you know that protein binds to DNA or DNA binds to protein? There are various experimental methods to do that. We will not be able to discuss each and every of these processes over here. But still, I just like to tell you the various processes which are available through which, if somebody wants to study some kind of protein DNA interactions, can go ahead. The first one is the electrophoretic mobility shift assay, or briefly known as EMSA. Another is the DNS1 footprinting. So DNS1 footprinting is basically a very interesting technique. So if the protein will bind to a particular DNA sequence, so that sequence will be hidden from the nucleases, and the specific nucleases which will bind there will not be able to attack them. So you have some kind of a assay system through which you can find out to which particular sequence now this particular DNA binding protein is binding. Okay, so there are others like exonuclease tree footprinting, then southwestern blotting, filter binding assay, ultraviolet cross-linking, and many chemical protection and reference assays. Amongst these, EMSA is the most frequently used because it is very, very simple, and also very, very sensitive method. So each of these processes have certain advantages and certain disadvantages. I will not, read this table and bore you, but you can see over here. For example, the filter binding assay is inexpensive and easy, but it has certain disadvantages. Okay, and similarly, if I go to the next slide, all of them have some kind of advantages, but also some kind of disadvantages. But EMSA, they are finding out, is almost a perfect technique where you don't have much disadvantages, or maybe this particular group may be actually unable to find it. But anyway, EMSA is very, very sensitive technique. We use it for our own aptamer detection work, and results are very, very convincing. Anyway, so various examples, where do you use them? Filter binding assay you use for identifying nucleic acid protein interactions, and similar. EMSA also identifying nucleic acid protein interaction, or you can also use it for identifying complexes in a cell, tissue extract, and so on. Right. So all of you know about gel electrophoresis. This slide is for people who don't know about these techniques and who have participated here. So gel electrophoresis is something you make with a polymer gel, and you make polymer gel with by cross-linking monomers. There are various monomers we use in our laboratories, like acrylamide, okay, and so on. Now, when we cross-link them, sorry, we don't cross-link them. First, we polymerize them. And during the polymerization, we also add certain cross-linkers, as a result of which you can get a fine gel structure. You can see here the pictorial representation. And on the other side, you can see the electron microscopy photograph of this particular gel on the top, right, which is a very soft jelly-like structure. But if you put it under electron microscopy, you can see these beautiful pores around this. Now, these pores have certain size limitations. It will allow certain molecules which are smaller than these pores to pass through them, and the bigger ones will be retained. So we use gel electrophoresis basically for separation purpose. This is something like the sieve we use in our home to separate the wheat flour or the rice flour from the rough or the bigger size particles. So this technique, gel electrophoresis, is used to separate mixtures of proteins as well as nucleic acids, but we use, say, agarose-gel electrophoresis to isolate bigger size DNA molecules, and then we use protein polyacrylamide gel for proteins, denatured proteins. We'll tell about the denatured and native gel electrophoresis. But anyway, the principle is very, very simple. You have this gel which you prepared in the first stage, and then you put it in a tank, and you have in the below picture you can see you load certain buffer who have certain particular pH, and you have a sample well. Now you add the samples and then you apply the electric field, and this electric field will drive the protein or the DNA molecule across this gel, and thereby separating them. So let us not go into much more details of these. This is a very simple technique known to all molecular biologists or life science people. So others, if you are interested, kindly read about this gel electrophoresis tonight. And even if you don't understand, you just need to remember that there is a process called gel electrophoresis through which you can separate protein and DNA molecules. Okay. Now, we basically separate the DNA molecule, protein molecules by opening up their tertiary or the quaternary structure back to their polymeric structure, if you remember the earlier discussion. So for doing this, we add a detergent called sodium dodecyl sulfate. And this kind of electrophoresis we call as SDS-phase electrophoretic mobility. Okay. Now, if we do not put the sodium dodecyl sulfate, the protein will not be denatured. Denaturation is something which is like boiling the egg and solidifying the protein. Okay. Of course, this is the most simple example I'm trying to give you. pH or certain kind of detergents will also help in the denaturation of a protein. So what in the case of native or non-denaturing gel, we do not use the sodium dodecyl sulfate, and the protein structure is remain, in its native state or intact, right? That means it still retains its function. So if we denature a protein, its function will be lost. But if we do not denature a protein, its function is still retained. For our particular case here, we are interested to retain the functionality of the protein to bind DNA molecules. Okay, that's why what we will do, we will use a native gel electrophoresis. Okay. Now there are other things like intrinsic charge of a protein, how these residual charge will help in its mobility. Let us not take all the trouble of remembering so many things at the same time. But let me focus on the native gel page. So this native page gel is carried out near neutral pH to avoid acid or alkaline denaturation of proteins, to study conformation, self-association or aggregation and binding of other proteins or compounds. Okay. So due to this native gel electrophoresis, we can use these for detecting many things like changes in charge due to chemical degradation, whether a protein is unfolded or molten globule, whether it is oligomeric and have aggregates. And most important and useful to us for this particular lecture, we can study binding events, whether one protein is binding to another protein, or a protein is binding to a ligand, or protein is binding to a DNA. So this is the principle of EMSA, through which we study DNA binding proteins, interaction of DNA binding proteins and DNA. Now you know about the gel electrophoresis, that you use electrical charge to push the charged molecules through the gel, which has pores, and thereby do the separation. Now, if you look in these pictures, these happy boys, it seems they are entering the school. I'm confused, or they may be exiting the school because it's now off time. Whatever, they are very happy, and they are very thrilled to, go either to the classroom or go to home. Now, probably they are going home or coming from home, right? They are not going from one classroom to another or from the classroom to the playground. Now, these same boys, these boys and girls are running with some speed, okay? Some of them are running faster, some of them are running slower, okay? And that's because of their intrinsic quality. That is how you also separate out the protein or the DNA molecules due to the intrinsic charges or the molecular weight, okay, or the molecular size. Now, next thing I would like to emphasize on is that these kids, if you ask them to run by removing these bags, okay, imagine that these kids do not have these bags and they are free and they start running. So under which case they will be running faster. So you can actually message me your answer. The kids will be running faster with this baggage or without the baggage. You can WhatsApp me somebody without bags. Okay, somebody told me without bags, right? Very good answer. That is exactly what I want from you. Now, tell me one thing. If we put a protein which is conjugated to a nucleic acid, because this protein binds to nucleic acids. So like these kids carrying the baggage, the protein is carrying a nucleic acid baggage, right? And the same protein without a nucleic acid baggage, and if we put it under electrophoresis, so which one will run faster? The protein with the nucleic acid baggage, or the protein or the protein alone? Which one will run faster? Protein alone will run faster, right? Okay. So now if you are convinced that the protein alone will run faster, then along with the protein nucleic acid baggage, then your concept of EMSA is clear. So we call it electrophoretic mobility shift assay. So there will be shift in the position of the protein having the nucleic acid baggage, which will run slower, then the protein alone, which will run faster. So that way we can, separate out, and we can also know that yes, these proteins are binding to DNA. Okay, now with this concept, let us now move slowly towards the DNA binding proteins, who these guys are, what they do, and where do you find them. Okay, now this is a figure which is representing the genome size on the x-axis and the protein coding genes and the y-axis. So a plot of genome size versus protein coding genes. And these are the various organisms from the various kingdoms: archaea, bacteria, eukaryota, virus. So virus is green colored, eukaryota, this one orange colored. I don't know what color you are, or orange or pink, orange color. And you have the bacteria, a little bit of dark green, and you have the archaea over here. So as we move from simpler forms of life, from the virus through the archaea, bacteria, to the eukaryota, you can see that the genome size is increasing, right? But of course, there are exceptions. There may be very many eukaryotes out here in below. You can see they have lesser, smaller genome size than the lower organisms. Okay. Now, more or less, you see that with the complexity of life, the complexity of the size of the genome is increasing. But this is not absolute truth. There are outliers over there. Okay. That is number one. Now, number two, with the increasing genome size, we can tell that the number of protein coding genes are also increasing. Okay, majority of the cases. But of course, again, there are outliers where you can see some of the eukaryotes, they have actually very big genome size, but their number of protein coding genes are very, very small. Okay, so these are the outliers. Let us forget about the outliers. We can go for the general trend. The majority trend is almost agreeing to this kind of a correlation. So this is a log-log plot, the genome size versus the protein coding genes. Okay. Now, out of these total number of protein coding genes, what is the size of the genome that codes for DNA binding protein? That's an interesting fact to know. We have around 2 to 3% of these protein coding genes in the prokaryotic genome, which codes for the DNA binding proteins, and this is almost double in the case of eukaryotes. 6 to 7% of the eukaryotic genome codes for DNA binding proteins. So this is a rough statistics you can always carry with you, that in humans you have roughly around 6 to 7 genes, percent of your total genes, which carry codes for the DNA binding proteins. Now these big number, it's a big number, 6 to 7%. So if you want to classify them, they will fall into one of the two classes, these DNA binding proteins. The first class is the proteins that binds to DNA in a sequence independent manner. Okay, non-specific binding. So you know one of the most important player in this class, the histones, which constitutes the nucleosome, and you know about the genome architecture or superassembly, how this DNA double helix wraps around the nucleosome core of these histone molecules. 8 of them comprise this core. So this is exactly what we are discussing today, DNA binding proteins. And this DNA binding protein is binding to the DNA in a sequence independent manner. These histones will not discriminate what DNA is coming to wrap around it. It will allow to wrap around it because it is not sequence specific. Now, all of you may be aware of this importance of DNA packaging. If our DNA would not have been packaged, this would have happened. I mean, our DNA, a person's DNA, when you unwrap it and stretch it, you can travel to the sun and back, not just 61 times. Okay, that's the number they have calculated out. I have never verified it, of course, but just to give some kind of excitement, that what is the importance of DNA binding proteins? If there would have been no DNA binding proteins, a single person would have to be so big, to accommodate its DNA within itself. I hope you can understand now, and also the importance of DNA binding proteins, how it helps in this packaging, which I mentioned you earlier. The next group is proteins that bind to DNA in a sequence dependent manner by recognizing specific sites in DNA. So this we call it site specific binding, like the binding of transcription factors or the restriction enzymes or endonucleases. And today we are going to discuss a lot about the second class. The first class we are not going to discuss anything because it's sequence independent or non-specific binding. A little bit of non-specific binding, of course, we will also discuss, but from a different context, not from this nucleosome concept. Right. Now, when it comes to DNA binding proteins. DNA may bind to, some of the proteins may bind to single-stranded DNA, right? And some of the proteins, they prefer to bind to double-stranded DNA. Now, let us start with the single-stranded binders. So the characteristic functional unit of these single-stranded binders is that they have a oligonucleotide or a oligosaccharide binding fold. Sorry. Yeah. Right. You can see this fold that facilitates the binding of the single-stranded DNA as well as various protein-protein interactions. These OB fold contain proteins. These OB fold containing proteins are essential for multiple aspects of genome stability and maintenance. And here you can see some of the proteins being shown. Now, what are the various work of these proteins? They are involved in the DNA repair. So this is a protein called replication protein A. Try to remember this, RPA. We may be using this protein in the next discussion. Then other things like replication fork modeling, and then you have checkpoint activation, and then also for telomere maintenance. So you see the single strand binding proteins have so many different kind of important role to play inside the cell. Now, this single strand binding family consists of two core subgroups. Number one is the simple SSBs, which contain one OB fold per polypeptide. And the other one is the higher order SSB, which contain multiple OB folds. Okay. And these eukaryotic RPA, which I was just highlighting in the earlier slide, is a heterotrimetric complex composed of 3 subunits, 70 kilodalton, 30 kilodalton, 40 kilodalton, referred to as RPA1, RPA2, and RPA3, respectively. Our genome, the human genome, encodes both these forms of simple and higher order SSBs. Okay. So try to remember the player RPA. Now we are trying to go and look into another protein which has also single strand binding domains. This is the famous protein BRCA2. Some of you may be familiar with it, with this protein. So you can actually text me the association of this particular protein with some kind of disease. Okay, so you know this already mentioned here. There's any mutation in this particular gene would lead to some kind of cancer susceptibility and chromosomal instability due to defects in the repair of double-strand DNA breaks. Okay, so that means BRCA2, which I'm telling has certain single-strand binding protein binding domains. Okay, is involved in the repair of double-strand breaks by homologous. Somebody is telling unable to join, Abdullah Jahangir. So Adiraz or John, kindly help this guy if he is. Others, you're all connected to me, right? Okay. Now, also Dr. Geeta Bharathi is unable to connect. Am I audible? Are we all audible? Let me check the number of participants. Yes. Okay. Thank you. Right. Okay. So these small technical problems will go on. Anyway, so let us see this particular protein, which is around 90 kilodalton, and this domain has 3 oligonucleotide binding folds. Okay, these OB folds we are discussing in the earlier case. It has 3 such folds and a helix turn helix motif, which binds to double-strand DNA. Okay, so this domain binds to single-stranded DNA, and this BRCA2 protein stimulates another protein called RAD51 and this mediates the recombination in vitro. They have found it out. Okay, so this is the BRCA2 protein, and you can see there are so many structural domains in the N terminus and in the C terminus. We will not discuss about these particular protein in detail because my idea here is to only show you 2 things. One is the cooperativity, another is the DNA binding domain. So we can focus on a few things like their BRC repeats. So they have 8 BRC repeats, which are located in the central portion of the protein, and these are primarily involved in binding to monomeric RAD51. The RAD51 association is very, very important. Okay, so and that is being done by through these 8 BRC repeats. Another important area is the password is incorrect. Dr. Geeta Bharti is telling, please help her. Okay, the C terminus of BRCA2 contains this TR2 domain, which interacts with RAD51 nucleofilaments. It also contains 2 distinct nuclear localization signals that are critical for BRCA2 nuclear localization. So this is another important area. And you can see here these OBB folds under these DNA binding domain. You have 2 domains. One is the okay, it shows password incorrect. So here this DNA binding domain is composed of a helical domain HD and 3 oligonucleotide oligosaccharide binding OB folds and also a tower domain. They promote BRCA2 binding to single-stranded DNA and poly-ADP ribose. This domain also associates with DSS1. So you see here, this particular BRCA protein has so many various functions. Out of these, you have these OB folds, and then which are binding to the single-stranded DNA. Okay. Let us try to move to the next slide. Now, this BRCA has a lot of biological functions. Number one is its involvement in homologous recombination. Okay, and here it do not act alone. It acts in a collaboration with many other players, right? The second function is, you can see it takes care of intra-strand cross-linking. So here you can see the blue strand and this orange strand. This would not have got cross-linked, this red one. So this is some kind of error, all right? Now this cross-linking actually will be broken by the BRCA2 involvement. You can see over here. So it repairs intra-strand cross-link repair. So these are all otherwise lead to some kind of mutation or genetic aberrations, right, at the molecular level. And then BRCA2 also plays a huge role in replication fork stability. So these are the various functional roles of BRCA2. Now let us move to another player, RPA. We have discussed about BRCA and also RAD51. Now this complex is called MRN. As the name suggests, M comes from MRE, R comes from RAD50, N comes from N51. These are various proteins, and they have various domains, like MRE has phosphoesterase domain, and this is a all of them are highly conserved proteins. And phosphoesterase domain is there, and there are 2 distinct C-terminal DNA binding domains. These DVD green ones, you can see they are the DNA binding domains. And this MRE possesses endo and exonuclease activity. That means it will chew DNA either from the center or from the sides. Okay. This RAD5150 is 100 kilo 50 Dalton. This enhances the affinity of MRE11 for DNA and stimulates its nuclease activity. So RAD50, if it binds to MRE, its nuclease activity will be enhanced, and its affinity for DNA will be stimulated. Otherwise, MRE11 itself doesn't prefer to bind to DNA, but once it is in a association, it has the DNA binding domains, but that character is stimulated by the association with RAD50. Now, this nuclease activity is actually further stimulated by the association of NBS1, which is a 65 to 80. This session is good. Can you have a recorded? Yeah, I think the Adira's recording is going on, right? Adira's kindly confirm. I asked you to do record. Yeah. Yes, thank you. So consists of an FHA domain and two adjacent BRCA domain here in its addition to an MRE interaction domain at its C terminus. So and we have ATM binding over here. This is a part of DNA damage response, and NBS1 is responsible for translocation of the mRNA complex into the nucleus in gene. Okay, now let us see how human SSB in the repair of double-strand breaks are there. So H as human SSB1, RPA, which I focused in the earlier slide, and BRCA2 are essential single-strand binding proteins in the repair of double-strand breaks by homologous recombinations. Now, this is a DNA double helix molecule, so this will get damaged. Now, this damage will actually be repaired by these players, SSB1, RPA, BRCA2. How? So, once this damage happens, the mRNA complex, which we discussed here, okay, MR11, RAD50, and BS1. So these entire complex, and they bind to one another, and the nuclease activity and DNA affinity of MRE11 is increased by the association of RAD50 and further by NBS1 that you already know. So when a DNA is damaged, right, this mRNA will be recruited over here in the site of the double-strand break by HSSB1. You can see this blue, small circles, right? So this is the SSB1. So the SSB1 will actually bring in this mRNA complex into this site. And you know that MRE11 has some kind of nuclease activity. So this will facilitate the resection of the 5' strand, okay? So it will start chewing the DNA from the 5' end, right? Now, these single-strand DNA stretches are rapidly bound by the next player, the RPA, okay? Which following BRCA2 binding, and after that, BRCA2 will be recruited. And after that, it is removed to allow for RAD51 nucleofilament formation. Okay, so you can now understand what is the importance of all these players we are discussing all the time. So their role in double-strand breakage, the role of SSB1, HSSB1, the mRNA complex, the RPA, and the BRCA2, and the RAD51. Okay. As a result of these, homologous recombination is facilitated by RAD51 mediated strand invasion into a sister chromatid. So if you go back to these slides, you can see this homologous recombination and strand invasion over here, right? How this RAD51 facilitated that. So I will actually give you the slides so you can study back these concepts we are trying to put across. So this double strand break repair, very complex phenomena or process. But okay, what I want you to carry home, the message is that it involves a lot of single strand binding proteins. Our focus here is absolutely from the nucleic acid binding protein point of view. The same thing you can look from different context. So today, since we are discussing about nucleic acid binding proteins, so the role of single strand nucleic acid binding proteins, very, very important in some of the fundamental processes of the cell. So this HSB1 has also been found to interact directly with RAD51 and to facilitate strand invasion in vitro. So this is additional information for all of you to remember. Now, so we have seen how these OBB or the DNA binding domains helpful, and how the complexation takes place, how one player after the other is recruited into one case study I took about the double strand breakage by homologous recombination. Now, let us move to the next point, protein DNA recognition mechanisms. So how does protein recognize DNA, either the sequence or the shape? Okay, so there are various mechanisms. Mainly, one is the sliding. So here you can see this pink ring. If you consider that as a protein, and this blue thread as DNA, so this ring is this blue thread is put through the ring, and this ring will move through the thread from one direction to the other. This we call as sliding. So the transcription factor here, represented by the pink ring, okay, or the protein, transcription factor means protein over here, move from one side to another by means of sliding. Okay, so it will go and stop at the side and check whether it has the sequences or the topology to bind to, right? If it finds it, it will stop over there. If it is not there, it will go to the next location. And this and then so on and so okay. But there are models where they tell it goes forward and reverse as well. Let us keep things very simple at this moment. Let us consider the unidirectional movement only. So this process is it's a continuous or process happening from one end to the other. So this process is in this process the protein is transferred from one base pair to another without dissociating from the DNA. Under ideal conditions, of course, the protein molecules are not rings like this. They are rather clamps, so they may open and then this is just to show the thing in a very simple way. What do you mean by sliding? The movement of a ring through a thread in a continuous manner from one point to another. In this case, we are discussing about the movement of a protein from one base pair to another continuously over a large distance. Okay, the second one is not so slow like this. The guy is impatient. It will, instead of, if it will bind to a DNA molecule. If it finds the site, fine, but if it doesn't find, it will not slide through. It will make a jump. Okay, that is what we call as hopping. Here, the transcription factor moves on the DNA by dissociation from one side and reassociating with another side. Okay. Now there is the third case called intersegmental transfer. Here the transcription factor gets transferred to DNA binding or the formation of a DNA loop. You can see this over here in the figure, resulting in the protein being bound transiently to both sides and subsequently moving from one on-site to the other. So these are the three mechanisms by which the protein DNA recognition takes place: the sliding, the hopping, and intersegmental transfer. Okay. Now, when it comes to the specificity, okay, how the specificity is, kind of implemented. I mean, how does the DNA will, protein will read the specific sequence or the topology that it is looking for. So there are two mechanisms for this. One is the base readout. You can see the blue box over here. The other is the shape readout. Okay. I believe most of you already know a lot about this base readout, and probably some of you know about the shape readout, or maybe all of you know about the shape readout. Anyway, so let me repeat what is a base readout. Take it as a small revision for you. So some proteins achieve DNA binding specificity through contacts with the bases in either the major or minor groups that recognize the base or base pair sequences. This type of recognition is often mediated by formation of hydrogen bonds between amino acids and bases, which convey the highest degree of specificity, and in some cases by water-mediated hydrogen bonds or hydrophobic contacts. In the case of shape readout, here for DNA binding proteins, where base readout cannot fully explain specificity, some of the proteins do not obey this base readout operating principle. So for them, we have to explain the phenomena through shape readout. particularly binding a non-ideal beta DNA bDNA factors like sequence-dependent DNA structures and deformability are proposed to play critical roles in this case. So these factors depend on deviations from the ideal bDNA and comprise a diverse set of mechanisms collectively called as safe readout. So these are the two differences in protein DNA specificity. Now, when we discuss about base readout, you may have a major groove base readout where the hydrogen bond, water mediation, and hydrophobic interactions comes into play. In the minor groove, hydrogen bond and hydrophobicity are important, but the water mediation is not important. Now, when it comes to the global or safe readout, you may have two kinds of safe readout. One is the local safe readout, okay, where the kings, the minor groups, and the major groups play a major role. And there's a global safe readout where the bending and the features of A DNA and Z DNA are very, very important. We'll try to discuss them briefly because we have many more points to discuss. So here, the base specific interactions in the major group comprises of hydrogen bonds with bases, and various examples here are the HTS domains. Okay, we will study about these HTS domains at a later point of time, and the zinc finger domains. Okay, and then immunoglobulin fold domains, then N-terminal end of basic leucine G-per domains or the max transcription factor. So these are the important examples when it comes to base readout. Now, there are base specific interactions in the minor groove as well. So here also the hydrogen bond formation and hydrophobic interactions are there, as I was telling you. And certain proteins like zinc finger proteins with cis to GATA-like domains form hydrogen bonds in the major groove, also bind in the minor groove. Okay. Then you have the high mobility group proteins from hydrogen bonds in the minor group, but rely on the recognition of DNA shape and flexibility to achieve specificity. So you see that base readout in a minor groove and base readout in the major groove are different. Just excuse me, Pulok, you are there? Okay. Hydrophobic contacts with bases. So here, some proteins contact only the minor groove, which often is associated with a dramatic widening and extensive hydrophobic content. So this mechanism is employed by TBP, SRY, and LEF1. The Tata box, TBP Tata box interface is completely dehydrated, and the abundance of hydrophobic contact in the interface suggests that they contribute to the specificity. So this hydrophobicity is also a factor when it comes to the specificity. Now let us move to the other type of specificity, which occurs due to safe readout. And as I already told you, both global and local readouts can contribute to DNA binding specificity. For local safe readout, such as minor groove narrowing, results suggest that the shape of the minor groove within a binding site can be read by a complementary set of basic side chains, most typically arginines when presented in the correct conformation. In contrast, global shape readouts such as a gradual band in the DNA helix may position elements of the DNA backbone such that these otherwise non-specific contacts, contacts can become highly specific. So when it comes to the local shape readout, here the small regions of 3 to 8 base pairs where the minor groove is narrow, and there are other things like DNA kings which are caused by the unsticking of a single base pairs. So here these two things lead to the deviation from the ideal bDNA. Now the conformation of double-stranded DNA depend on many things. One is the ionic or hydration environment. So low hydration environment will favor the A DNA. High salt results in the Z DNA. Second, important thing is the sequence. A DNA favored by certain stretches of purines or pyrimidines alternating purine pyrimidine favors ZDNA. Then certain proteins bind to DNA in one helical conformation and force conversion to a different conformation. So for example, a protein may bind to a DNA and later on convert it into ZDNA or the vice versa. Okay. So these are the various conformations of DNA, ZDNA, vDNA, aDNA, vDNA, and ZDNA. All of you know about them more or less, or these are very simple things taught in high school. Maybe you have forgotten. ZDNA is, helix sense is left-handed, and it has more base pairs. Okay, and then you know that jet names comes because of this zigzag nature. Now, when you look into the molecular shape and electrostatic potential of A, B, and Z DNA, they differ. So you can see the green figures over here on the top, A, B, C, D. So you have A DNA where the GC content is more, and B DNA where the GC content is more, and you have a B DNA where the AT content is more, and you have a Z DNA where the GC content is more. And you can see these very interesting features in the major groove and the minor groove. You can see you can just see the figures and appreciate the differences. Okay, and you have here in this topology, certain concave features, right, in the groove, and the green one is the gray one is the concave surface, and this green one is the convex surface. Okay, and you can see that they are different for each form of the DNA. On the top, you see these images of the 3 helical forms of DNA containing alternating sequences of around 7 mers. And the bottom, you see the variation of the electrostatic potential at the molecular surface due to the shape in the atomic charges. So the red one reflects the negative potential, the white one the neutral potential, and the blue one the positive potential. So you can see for the same sequence, right, alternating sequence, this GC 7 times, right, the electrostatic potential on these molecules are different, right? So this shape as well as this electrostatic potential, right, governs the binding of DNA to this molecule. This is what I want to highlight at the end of this slide. And the shape specificity comes from this kind of primary phenomena associated with the DNA topology. So you can see here the minor groove shape over here. There's a narrow minor groove, and there's a narrow minor groove electrostatics. You can see over here. The narrow region of the minor groove that is contacted by arginines here is highlighted in the blue. Okay, and the right, you see the electrostatic potential of the operator calculated in the absence of the repressor, and this is plotted on the molecular surface. Now you have here one molecule which is SCR specific. This is FKH 250 under figure C on the right hand, you can see, and a Hawkes consensus FKH 250 con. So here the binding sites differ in minor groove shape, a structural feature intrinsic to these sequences. These local variations in shape result in an enhancement of negative electrostatic potential at distinct positions that attracts arginines into the minor grooves. Okay, and you can see here in the minor groove under figure C, you can see the negative charges over there. But in under figure D, FKH 250 consensus, you see there is no any such negative charge accumulation over here. The SCR N-terminal arm uses this sequence-dependent variation in shape and electrostatic potential to achieve the DNA binding specificity. Since the narrow minor groups are often associated with AT-rich sequences, enhancement of negative electrostatic potential in the minor groove which in turn is recognized by arginines, offer a general mechanism for sequence-specific recognition of the DNA chain. Okay. Now let us go to the next slide. So here we are focusing again on the minor group shape. Okay. Here Hox DNA binding specificity is mediated by local shape recognition. We are discussing about local shape readout. So here we have two panels, SCR specific FKH 250 and a Hox consensus FKH 250 concentration consensus binding site B. Okay, then this FKH 250 has two minor group minima, which creates a more negative electrostatic potential, which I already told you in the earlier figure. And here, the W, you can see here, W refers to the Hox YPWM motif that makes a direct contact with the cofactor EXD. Now, what happens in situation A? Here you see SCR no EXD, not stable, figure A. So in the absence of EXD, SCR does not bind with high affinity to FKH 250 because the arginines of the N-terminal arm and linker of SCR not positioned correctly. Okay, because here there is no EXD. EXD is this green molecule below, and on the left side you can see in the other three figures you can see the presence of EXD. When there is no EXD, this binding will be not there because the N-terminal arm and linker of the SCR are not positioned correctly. Okay. Now, under condition B, in the case of Hox proteins, here also it do not bind well to FKH 250 even in the presence of EXD because their N-terminal arms and linker regions do not have the correct residues. Okay. So here also binding is not taking place properly. Now, when it comes to, on the situation C, so what is happening in C? SCR and EXD heterodimer both are present and they bind well to the FKH 250 because the SCR N-terminal arm and the linker region have the correct residues, and EXD positions them correctly by binding the YPWM motif. So this case, the binding is successful because everything has fallen into place and everything is favorable over here. Now, in the fourth case, what is happening? The other Hox EXD heterodimers bind well to FKS 250 consensus. Okay, here you don't have any negative charge over here, but this FKS 250 consensus is there. So due to these, the binding is happening because this binding site is not as selective because it has a less negative electrostatic potential. Thus, the sequences of the Hox and terminal arms and the linker regions are not as important for binding in this particular case. So there are various examples of local shape readout, but let us stop at the, with this particular example. Let us try to go for the global shape readout, where you have important factors like the bending and even the A-DNA and Z-DNA topology. We'll not discuss the A-DNA and Z-DNA topology again and again. In this figure, you can see with the same sequences, the topology of A and Z-DNA varies, and it is from here you can make out that this will facilitate different kind of global shape readout. Okay, now let us take only one example regarding the bending, how this is important. So here, some DNA molecule, protein molecules will bind to DNA and form bands. Okay, now what happens to SADs? They have developed flat assays where you have a donor D and an acceptor molecule, and they cause energy transfer. right? And then if a protein binds to to a particular portion of this probe, the acceptor and donor will come closer to each other, as you can see in this figure on the left side below. And due to these, the energy transfer would occur, and we can detect them very easily. So we take advantage of these phenomena to study the bending of DNA due to protein binding. So there are various examples. I'm just like to give you only one. Here you can see one protein called ISF DNA. ISF. This ISF and binds to DNA, and this is a crystal structure they have solved. This is showing the protein subunits in pink, which are beta, and white, and the intercalating prolines in the yellow. So DNA is color coded according to the solvent accessible surface revealed by the hydroxyl radical footprint in solution. Green, light blue, and dark blue, they indicate mild, moderate, and strong protection from cleavage respectively. And yellow, orange, and red indicate mild, moderate, and strong enhancement of cleavage. The two segments of DNA formed symmetry-related complexes within the crystal are included at the bottom to accommodate the full ISF footprint. So I told you about DNA footprinting. So here, since the protein is getting bound over here, so this particular portion will be, immune to any kind of nucleus degradation. And you can see here two arrow marks, right? So these are the attachment position of the donor and acceptor fluorophores, and these are attached over here. So you can see this structure of the ISF bound to the A site from the bacteriophage DNA. So here we have this DNA substrate that contains one of the cognate sites, A site from the base pair 15 to 49 of fast DNA, and that binds specifically to E. coli integration host factor. ISF is a multi-purpose architectural protein which plays an essential role in several cellular processes, including site-specific recombination, transcription, and DNA replication. Now, here we discuss about the structure of GPNE1 DBD and model for cooperative GPNE1 ISF DNA binding. So here figure A is the high resolution NMR structure of the DNA binding domain shown in the ribbon representation. This is a winged HTS motif, and in figure B you have the cartoon depiction of cooperative assembly of GPNE1 and ISF at cos B. The GPNE1 dimer blue binds to two R elements, the R3 and R2 spatially separated in a duplex, and the duplex bending is supported by IHF, which is purple in color. So here you can see the ISF E. coli integration host factor, and in figure C you see the molecular model showing the cooperative binding of these molecules, GPNE1 and ISF. The R elements in the DNA duplex are colored green and yellow. The alpha and beta subunits of ISF are bound at 1 are colored blue cyan. The GPNE1 dimer are colored blue cyan with wings and recognition helices colored purple and red respectively. So this model for cooperativity and these particular experimental designs helped us to understand how a protein molecule, when it binds to a DNA, can cause a band. And you can see all these experimental details in these papers in the molecular cell. My purpose here is to just tell you how the DNA binding, protein binding to a DNA changes its molecular structure. We'll try to avoid the discussion on aDNA and ZDNA because of the shortage of time. And then I'd like to go to a most important topic in this lecture, the taxonomy of DNA binding proteins. So we know that roughly around 6 to 7% of the proteins that is there in the eukaryotic genome belongs to the class of DNA binding proteins. And we have a huge several thousands of proteins. So if we calculate out the total number of the DNA binding proteins, leaving aside the nuclear binding proteins, where you have again the RNA binding proteins as well as both DNA and RNA binding proteins, the number is quite huge. So it is better to arrange them in under certain classes for ease of for academic purpose as well as for scientific purposes. So in 1990, the first taxonomy of DNA binding protein was proposed by Harrison. 5 years later, it was revised by Luisi. And again, in 2000, it was revised by Luscombe et al. And which every revision, the refinement has been done. So I am trying to present here the final, the last revision by 2000, in 2000 by Luscombe et al., where their basis of taxonomic arrangement was that the members of the same group share a prominent structural feature used for DNA recognition. Okay, so by this time we right now know how protein recognizes DNA. It's a sequence specific, or it may be shape readout, or it may be base readout, right? And there are many other things, not possible to cover everything in one single lecture, but we know that there are structural features in DNA which are used by the proteins for identifying them or recognizing them. So using these structural features, Luscombe et al. And his prior workers did the taxonomic classification of the DNA binding proteins and found out that they belong to 8 different groups, like the helix turn helix proteins, which also include the winged helix turn helix proteins. Then you have the gene coordinating proteins, you have the zipper types, you have other alpha helix binding proteins, then the beta sheet binders, the beta hairpin and ribbon binders. Then there are some other miscellaneous types. And finally, the enzyme types, the 8th class. The enzyme type, of course, is not very structurally similar to one another, and it may contain many proteins which are already being classed under the other 8 proteins, other seven groups. Okay, so anyway, this is the statistics of their classification, Luscombe's classification. So you have 8 different protein groups, 1 to 8, from helix turn helix to enzymes, right? And you can see each of these groups have a certain number of families. The largest families belong to helix turn helix, roughly around 16, and to the enzymes, same 16. And then you can see the distribution of these DNA binding proteins across prokaryotes and eukaryotes. In helix turn helix, you have 32 number of proteins under the prokaryotes and 28 in eukaryotes. This doesn't mean that these proteins are much more dominant in prokaryotes. This is because the data has been taken from the available databases. So as studies progress in future with more organisms, this statistics is going to change. So total you have around 60 members of helix turns helix, 23 gene coordinating, 10 zipper types, other helix 4, 8, beta sheet 8, beta hair ribbon 11, others 8, and enzymes you see the largest family 113. But this is a very, very heterogeneous family. And total you have around 241 proteins, right? Okay. So just if you take the example of the helix turn helix group, you can see the various 16 number of families, the Crow and repressor family, homoiodobin family, LAC1 repressor, endonucleases, and so on, right? transposes, catabolite, then transcription factor family, ETS domain, and so on. Now, let us quickly see what is a helix turn helix protein binding motif. So this motif is a common recognition element used by the transcription regulators and enzymes of prokaryotes and eukaryotes both. So it has 20 amino acid segment of two almost perpendicular alpha helices. So you can see the blue and the yellow one. So they are perpendicular to one another. And these helices are very small ones, actually, only just 20 amino acid long. And they lie at 90 degree to one another in three-dimensional space. And this HTF motif bind in the DNA major groove. So in the beginning, I was highlighting the DNA topology, how the major groove and the minor groove arise. Okay, if you remember it. So starting from that point up to this point, and also the topology of A DNA, B DNA, and Z DNA, you can well appreciate the role of importance of these groups over here. And HTH motif specifically binds to the DNA major groove. And the second alpha helix is commonly known as the recognition helix or the probe helix, is inserted in a red groove. Here, if you go to the original paper, you will find out they are depicting this red portion as the portion who is binding to the major groove. Now, under this class HTH group, you have several enzymes. Okay, this is a transcription factor. Okay, but you have several enzymes. So out of them, one of the most, there are many like that, heme recombinase, gamma delta resolvase, and one enzyme that I would like to highlight here is the FOC1 restriction endonuclease. This FOC1 restriction endonuclease is important from the context of this workshop where we are going to study about genome editing or genome engineering, and there is going to be a lecture on two technologies, the ZFN and the TALEN, where this enzyme plays an important role. So I'm not going to tell about the properties of this enzyme. You will come to know about the properties of this enzyme by tomorrow in another lectures. So under this HTS, you have one special class called winged HTH. The name winged comes because if you go into this structure, it's a very simple structure of 20 amino acids, right? But here you have a much more elaborate and majestic and a complex structure. So how does this structure happen? It happens with the presence of a third alpha helix and an adjacent beta sheet, which are considered to be components of the DNA binding motif. So recognition is similar to the regular HTM motifs. You can see here it is binding to the major group. There is red portion, but the extra secondary structural elements provide additional context with the DNA backbone. Okay, so you have another second group is the gene coordinating protein. So gene coordinating protein is a generic term which is used for all proteins having zinc ions in the DNA binding motif. The gene coordinating proteins comprise the largest single group of transcription factors in the eukaryotic genomes. Now, I just like to draw your attention to these particular sequences on human, Genopus, eastern mouse, and you can see here the yellow colored CC and the blue colored HH. So remember, these sequences have some interesting motif where two cysteines and two histidines are there, right? And they play an important critical role over here. So here, if you go to the left side, you see the three zinc finger motifs found with the major group of DNA strand with a single zinc finger motif being highlighted in the black box. The ZN2+, which is purple colored, is bound to two cysteine and two histidine ligands within an alpha helix and anti-parallel beta sheet. Okay, so please focus on this particular diagram over there, and the reason why I asked you to pay your attention to the two cysteine residues and the two histidine residues, cysteine residues and histidine residues in the DNA sequences to which this particular protein binds. Okay, so on the right side you see the DNA binding motif is characterized by the tetrahedral coordination of one or two zinc ions. Okay, by conserved cysteine and histidine residues, the metal ions are believed to provide structural stability to the domains that are not sufficiently large for a stable hydrophobic core. So this gene here plays an important role. That's why you call them as zinc coordinating proteins. Okay. Now there is another term called zinc finger. So to avoid this confusion, term zinc finger is used for proteins that have the ZIF 268 style motif with two beta strands and an alpha helix. So this particular protein is a little bit different from the other one. So this is the third group, the zipper type proteins. So you have the two families. One is the leucine zipper, and the other is the helix loop helix proteins. And you can see this figure on the right, the yellow coil and the blue coil. You see a portion which is binding to the DNA, and you see another portion which is close to one another. That portion is the portion which is the dimerization module or the dimerization zone, and the other area is the DNA binding zone. This is a very interesting. So from the name, from the shape, you see it appears like a zipper, and hence the name. So the DNA binding region is found in the amino terminus, and the binding segment is a direct extension of the dimerization region. Okay, so it's a continuous. I mean, a protein, and there is no any separation between dimerization zone and DNA binding as such. But of course, one portion dimerizes with the other polypeptide, and the other portion, remaining portion, binds to the DNA. The alpha helices of the two subunits diverge from the coiled coil and enter the major groove. So it's a major groove binder in opposing directions. So it will almost embrace, it will embrace the DNA molecule, each binding to half of the target DNA. So this is the first family under the zipper type proteins, the leucine zipper. And there is another called the helix loop helix proteins. So it is similar like the leucine zipper proteins. But here, between the dimerization module and the DNA binding module, there is a presence of a loop. Okay, so here there is a clear-cut differentiation of the two functions, the dimerization function and the DNA function. And remaining things, the binding is almost similar. Okay, I hope you now understand the difference between the leucine zipper proteins and the helix loop helix proteins. So there is a group 5 and group 6. We are trying to studying them together due to shortage of time. The group 5 is the beta sheet protein, and the group 6 is the beta hairpin ribbon proteins. Okay, they are almost similar but yet different. So both these groups contain proteins that use beta strand structures. All the time we are discussing about the alpha helices, right? How the alpha helices bind to the major groove. Okay, now here these are the players are different. It is the beta strand structures which are being used for DNA recognition and binding, and they fall under group 5 and group 6. So let's start with the group 5 first. This contains the Tata box binding protein family and is characterized by the use of a wide beta sheet to bind the DNA. These proteins are an essential component of multi-protein transcription initiator complexes that assemble or promoters of genes transcribed by RNA polymerase 2. Now, what are these group 6 proteins? They are almost same like the group 5 proteins, but they differ from the Tata box binding proteins in that they use smaller 2 or 3 stranded beta sheets or hairpin motifs to bind in either the DNA major group or minor groups. So you can see here the yellow, you can see here they are using hairpin motifs over here. Okay, so this is the fundamental difference between these two groups. Otherwise, they are all beta sheet structures, beta strand structures, which are utilized for the recognition of DNA. I avoided discussion of the other group, but and straight away jump. Just one group I left, the group 7, because of time management. So we are going to the last group, the enzymes. Okay, and the enzymes are the highest numbers, as I told you in the beginning in the statistical table I showed you. The enzyme group do not have a common structural motif for DNA binding. and are included on the basis of their functions. Okay, the function is that these enzymes alter DNA structure through the catalysis of a chemical process and use an extensive combination of helicases, beta strands, and loops to recognize and bind DNA. There is no any common structural motif, neither any common mechanism. They may deploy anything, the alpha helices, the beta strands, or the loops to bind to the DNA. So enzymes bind to DNA by all type of available means, right? Many enzymes comprises 3 distinct domains. Okay, in spite of this, we can have some generalities. Number 1 is that a DNA recognition domain that reads the DNA sequence. Second is a catalytic domain with the enzymatic active site, and where applicable, a dimerization domain. Although clearly there are exceptions. Okay, these are some of the common things, but there are also exceptions because the enzymes do not have something structural or functional. Functional thing is common. They will alter the DNA, but then this DNA alteration is again also of different kind. Some may cleave the DNA, some may phosphorylate the DNA, and so on. Okay, and then many of these enzymes actually fall under various classes like HTAs or other groups. And then some of these you will come to know are important from the genome engineering and genome editing point of view. The resulting structure often has a U-shaped activity cavity in which for all these enzymes you will find a U-shaped cavity in which the DNA is bound, and often the DNA structure is severely deformed on binding. Okay, so this kind of mechanism is common to this class. For sequence specific enzymes, the target sequences are typically 4 to 8 base pairs long, and binding is far more discriminating than that of the transcription regulators. Often a single change in the target sequence can lead to over a million-fold reduction in binding affinity. So these we know about the restriction enzymes, right? I mean, any base change, they may not be able to recognize it. That is why you do the restriction fragment, polymorphism, RFLP, and all those things are depend on this kind of biological phenomena. The proteins are thought to derive the specificity from both readout of the base sequence and the catalytic action of the DNA, as in endonuclease MAMH1 and EcoR1, or even primarily from the catalytic process, as in endonuclease EcoRV. There are other proteins such as polymerases. They must, however, provide sequence-independent interactions with their DNA substrate, yet retain the specificity to distinguish correct paired bases from the mismatches. So 7 endonucleases and 4 polymerases dominate this group of 16 family. So this is the 16-member family of the enzymes ranging from 39 to 54. And you can see the dominating members are the endonucleases 40, 41, 42, 43, 44, 45, 46. You can see endonuclease PVO2, so on and so forth, DNase 1, DNA mismatch endonuclease. These are the, I think, 7 members, right? Endonuclease is 7. And then you have again 47, 48, 49, 50. So these are the polymerases, okay? Out of this total 16, so the dominating members in the class enzymes are the 7 endonucleases and the 4 polymerases. And you have this PDB code of these particular enzymes and the protein name over here, and from the species from where they obtained, the DNA sequence on which they act. So finally, thank you. So my lecture was supposed to end at 4:45. I just could make it at 4:41. So if you have some queries, we can go to the discussion briefly. And our speaker panelist is already there from USA. He has been staying awake in the early morning. So yes, some questions if you have, you can put in the chat box. I can see Suresh sir now. Yeah. Hello, sir. Yeah, there's a question from Hema Jagadishan. Thank you for the detailed explanation. Can you predict the probable sites of binding of the DNA binding proteins, especially TFs, and predict gene expression based on the TF expression, especially when multiple TFs regulate gene expression. Yeah, a lot of bioinformatics work has already been done. And in fact, some of the things that I was telling you was actually, predictions, right? So several groups across the world has worked. I mean, this is not new, something actually quite old. We can do this, yes. Any other questions? Okay, thank you.